Symptoms that you see manifest are a direct result of the activated acute inflammatory response. The acute inflammatory response becomes activated when tissue damage occurs, whether this is through injury such as a cut, insult such as an infection, or trauma such as a sterile injury like a burn. Either way, this activates mast cells and tissue resident cells in the inflamed area to secrete histamines and leukotrienes that serve the purpose of causing swelling so that this inflammation remains in the local environment. At the same time, macrophages in the area become activated and damaged epithelial cells both begin to secrete large amounts of pro-inflammatory mediators, including interferon gamma and IL-6, all of which serve the purpose of recruiting our immune cells. Primarily in our acute inflammatory response, these are polymorphonuclear leukocytes. They have been shown to be the first cells to arrive in the inflamed area and therefore have been shown to help accomplish the overall goal of acute inflammation, which is to rapidly remove the extracellular insult. So I want to talk a little bit briefly about polymorphonuclear leukocytes, PMN, or you may have heard them called neutrophil as well. These cells are polymorphonuclear, right? So they have poly means multiple nuclei, as you can see here, these three lobes. And they also are granulocytic, which means that they possess all of these granules with different cytotoxic enzymes that help in their effector functions. PMN comprise approximately 50 to 60% of total circulating leukocytes in healthy individuals, with these numbers increasing to approximately 80% under inflammatory conditions. They can be identified in studies by the expression of mature myeloid markers, lymphocyte antigens 6C and 6G, and have been shown, their infiltration and response specifically have been shown to be required for initiation and resolution of acute inflammation. So before these cells can play their role in acute inflammation, they must first be recruited from the bone marrow. For this to happen, the CXC motif receptor 2, or CXCR2, on the surface of PMN must be activated. Under healthy conditions, PMN are maintained in the bone marrow by CXCR4 mediated retention signaling. This is perpetuated by the continuous secretion of CXCL12, its ligand, by the bone marrow stromal cells. Under acute inflammatory conditions, high expression of chemokines CXCL1, 2, and 8 in the serum can bind the CXCR2 receptor, which is their receptor, on the surface of PMN in the bone marrow. This binding inhibits that CXCR4 retention signaling and increases the expression of CXCR2 on the surface of PMN. CXCR2 is also important for their activation once they arrive in the tissue, so in this way it helps them prepare. This ligation also induces expression of adhesion molecules that are required for PMN to exit into the bone marrow. Exit into the circulation. Once in circulation, PMN are tethered at the site of inflammation to the endothelium by binding and releasing, weakly binding and releasing selectins on the endothelium surface. This allows PMN to initiate rolling, which allows them to get activated by interacting with molecules on the surface, trapped on the surface of the endothelial cells. This activation induces expression of high affinity adhesion molecules, LFA1 and MAC1, so CD18 and CD11B, and for that firm adhesion to the endothelial membrane. Once firmly adhered, they use their chemosensory abilities to crawl towards the site of infiltration or the site of inflammation using a chemotractic gradient. Usually they decide where they want to infiltrate by the high concentration of chemokines at the area. Once they find this high concentration of chemokines, adhesion junction proteins between the endothelial cells help to mediate their paracellular, which means between cells, transmigration into the tissues. When a PMN arrive in the tissues, they uptake and degrade pathogen in a process called phagocytosis. Phagocytosis further induces neutrophil to release cytotoxic enzymes, including myeloperoxidase, neutrophil elastase, MMPs, and ROS, that all act to damage proteins, DNAs, and lipids of pathogens and infected cells. They also have been recently shown to produce neutrophil extracellular nets. These are chromatin fiber nets that are coated with these cytotoxic proteins that neutrophil put out to catch or to immobilize extracellular pathogens until they can be phagocytosed. While neutrophil effector functions are very important, they are overall important or required for the resolution of acute inflammation. Specifically, resolution of acute inflammation depends on PMN clearance of pathogen, damaged cells and infected cells, monocyte infiltration and maturation, apoptosis of PMN, and macrophage uptake of these apoptotic PMN. Macrophage uptake of apoptotic PMN has been shown to induce a plasticity switch to the M2 phenotype, which has been reported in literature to help facilitate the resolution of inflammation by secreting growth factors that recruit fibroblasts and help to rebuild or repair blood vessels. So, in amendment to my earlier goal of inflammation, the goal of acute inflammation is to rapidly remove the extracellular insult, suppress the immune response, allow for tissue repair, and return to homeostasis. However, in conditions where the resolution stage is disrupted or there's a failure, it results in chronic or post-acute inflammation. And while the causes of chronic inflammation differ between the individual, genetic susceptibility, immune system dysregulation, and environmental factors have all been shown to be contributors to this progression. PMN infiltration and response has also been shown to significantly contribute to chronic inflammation development, specifically through bystander tissue damage effects, where those cytotoxic enzymes damage the tissue that is actually trying to heal, decreasing epithelial membrane integrity. Their mass infiltration of the submucosa damages those proteins, those junction proteins, and then perpetuating leukocyte infiltration. They secrete stimulatory factors that recruit other leukocytes as well. Therefore, PMN have been associated with the pathology of various chronic inflammatory disorders such as rheumatoid arthritis, Crohn's disease, and ulcerative colitis. Chronic inflammatory conditions have also been shown, PMN have been shown to be regulated alternatively under chronic inflammatory conditions. Studies show that PMN demonstrate enhanced phagocytosis, prolonged lifespan, which leads to an exacerbated immune response, because remember, again, their death and their clearance is needed to move into that resolution phase. Continuous production of these cytotoxic enzymes, which further contribute to tissue damage. The ROS also helps to promote infiltration of other immune cells. And net formation specifically has been associated with thrombosis, small vein thrombosis, which can cause leakage of blood into the tissue. So, unfortunately, loss or inhibition of these functions also can result in chronic inflammation. So we need novel methods to regulate their recruitment and response under these conditions. In a study by Vienna et al., they begin to investigate how neutrophil recruitment or infiltration was regulated under chronic inflammatory conditions. To do this, they investigated, they used thymosin-induced peritonitis as a in vivo PMN response assay. Specifically, healthy black 6 mice, approximately 20 grams, were IP injected with thymosin, or intraperitoneally injected with thymosin, and then they looked at the peritoneal cavity at multiple time points between 2 and 8 hours. After allowing these cells to infiltrate for 2 to 8 hours, they expected to see this infiltration. And then they collected the cells by peritoneal lavage after euthanasia and labeled them fluorescently for markers for PMN or neutrophil. Here in this figure, they show that beginning at 2 hours, we see some infiltration, but between 4 to 6 hours in these mice, we see massive infiltration of PMN, and this infiltration slowed or came to a stop around 8 hours when these numbers started to decrease in the peritoneal cavity. Along with this, they used a similar model, but instead of healthy mice, they used chronic inflammatory mice or inflammatory mice. For my purposes, I'll talk about the DSS model because that's the model I use in my paper. They also looked at this in diabetes models, lung inflammation models, and cancer models. But in their DSS model, when DSS mice were injected with thymosin, they found that these cells demonstrated enhanced responses arriving mostly, about 80% cells arriving in the peritoneum at 2 hours when compared to those healthy cells that arrived at 4 hours. So when they got this result, they wanted to figure out if this was a result of the environment or if these cells are actually changed functionally so that they arrive earlier. They looked at, they conducted adoptive transfer experiments. For my purposes, I'll focus on the one where they looked at the effects of the cell versus the environment. To do this, PMN were isolated from colitis mice and adoptively transferred into healthy mice that then received IP injections of zymosan. These mice were allowed to go for zymosan peritonitis was allowed to go for 2 to 4 hours in this case because they had already identified this to be the best time for that infiltration. These mice were euthanized and their cells were collected by peritoneal lavage and they were fluorescently stained. They found that when cells or PMN from mice with inflammation were transferred into healthy mice, these cells still arrived in the peritoneum earlier than their healthy counterparts. So this finding is really what helped to discern my dissertation project. The major takeaway from this for my project was that they found that these functional changes were displayed prior to PMN release from the bone marrow, because in that adoptive transfer experiment, they took those cells directly from the bone marrow. So their infiltration suggests that they're already changed before they exit into circulation. But it did leave some questions, such as, when a healthy mouse becomes inflamed, is there a reprogramming event that truly occurs? What is the functional significance of this reprogramming? What factors are associated, and do PMN remain altered as this inflammation is resolved? So to investigate these, I created the following aim, which was to investigate the reprogramming of the bone marrow myeloid compartment under chronic inflammatory conditions. To do that, I plan to analyze the bone marrow myeloid compartment for population changes, examine phenotypic and functional differences in the population, and identify a factor or factors that may be regulating these changes. To do this, we created a DSS-induced colitis to recovery model in which mice were treated with 2% DSS in water. These were healthy black 6 female mice, approximately 20 grams in weight. They were treated with 2% DSS in water for 9 days. And while the mechanism of DSS induction is still up for debate, it is commonly agreed upon that the DSS irritates the epithelial barrier, which creates holes through damaging tight junction proteins that allow bacteria, commensal bacteria that normally reside in your lumen, to enter into the lamina propria and activate tissue-resident cells to secrete large amounts of pro-inflammatory mediators. These are the same mediators that we need to recruit our target cell out of the circulation and into the inflamed area. And as I previously mentioned, these cells are really wonderful at removing insult, but they have serious detrimental effects. In my model to look at these changes under resolution, we removed this insult and allowed these mice to recover until day 30. Just a bit on DSS by itself as a model. Like I said earlier, we decided to use this model because it showed the greatest changes in the original paper. But as a model itself, it has been shown to mimic the human ulcerative colitis, which is known to be, or PMN associated. Which is known to be PMN associated. And overall, its rate of induction, simplicity, and reproducibility made it a really good model, or efficient model to use for this study. So using this model, we were able to successfully induce colitis in our mice, evidenced here by decreased body weight, increases in disease activity index, and increases in colon tissue loss, all at the post-acute, beginning around 9 days, or chronic stage of treatment, with all of these returning to normal when the insult was removed. Additionally, we looked at IL-17 expression in the serum, and we looked at cytokine expression in the serum, and we saw that our findings recapitulated other studies that saw that IL-17 was the primary cytokine systemically upregulated during colitis in mice. We looked at fecal lipocalin expression too, as it has recently emerged as a biomarker of both inflammation, and resolution, where it is secreted during inflammation by mature degranulating neutrophils, and during tissue repair by the damaged and repairing epithelial cells. And indeed, our studies show that during acute or post-acute inflammation, we have increased, twofold increase, and that is further increased under repair conditions after the insult was removed. All taken together suggesting that our model successfully established colitis and mimicked the subsequent resolution phase we wanted to study. After we established that our model was actually what we wanted to use, we then began to tackle that first thing, and that was to look at the changes in the bone marrow myeloid population. To do this, I used a modified Kirkhoff gradient that was coined in my lab in Dr. Yuanlu's laboratory, where Kirkhoff at percentage 5, 40, 50, 60, and 70 were layered to help isolate our total bone marrow myeloid compartment. To do this, our treatment mice were euthanized, and whole bone marrow was collected by flushing from the femur and tibia, and they were loaded onto the top fraction of this gradient. The gradient was then centrifuged, and we get separation of 4 distinct density decreasing bands, which from previous studies we know fraction 1 contains stromal cells and fibroblasts, fraction 2 contains B lymphocytes, but for this study we were interested in these last 2 fractions, which we show here contain a mixture. Fraction 3 contains a mixture of immature granulocytes and monocytes, and fraction 4 contains primarily mature PMN. And then here we just overlay them again so that you can see the differences between their cell surface size and complexity. So now that we identified and made sure that we were getting the correct population, we conducted this experiment at various time points throughout the treatment. We found that just when we were separating out the immature myeloid compartment, we had expansion of primarily granulocytes in that compartment. Granulocytes in this compartment. We also noticed that they show slight changes in their cell complexity during this time too, which suggests that these cells are expanding under these conditions. So to look at their expansion in a whole compared to the other compartments of the myeloid of the bone marrow, we collected each fraction, counted it, and expressed it as a fraction of or a percentage of the whole to create this data. And basically, we found that chronic inflammation induced significant expansion of the immature myeloid compartment, specifically between days 9 with this returning to baseline at day 18, and that at the same time we see decreases in the PMN bone marrow population. These decreases were likely a result of their rapid recruitment or their massive recruitment into the inflamed tissue. So after seeing these changes, so that would be the first part we wanted to look at. So after seeing these changes and seeing that, being surprised that the actual immature compartment expand, we decided to look at the phenotypic and functional changes of both the immature and the mature myeloid populations. So in our mature PMN population, we again isolated our PMN from fraction 4, and we activated these cells with PMA so that they would begin to secrete those reactive oxygen species. We see that, again, during the post-acute phase of treatment, they already have high expression of these cells compared to the immature cells, but they further increase their ROS production during post-acute colitis. And for our next piece of data, I just want to explain how we did this. We did adoptive transfer experiments where fraction 4 cells, so our mature PMN cells from DSS mice, were IV injected into healthy mice, similar to the experiment I explained earlier, and zymosan peritonitis was induced for 2.5 hours, and these cells were collected by peritoneal lavage. These results showed that PMN also display enhanced infiltration capacity during the post-acute or chronic phase of this treatment. as well as increased pathogen killing capabilities. When we looked at some of these functions, we looked at characteristics of mature neutrophils. So we wanted to look at the CXCR2. Remember I said these receptors needed for migration out of the bone marrow for mature neutrophils. In general, immature neutrophils do not express CXCR2 or express low levels of CXCR2. We found that during the post-acute phase and lasting throughout the resolution phase after the insult was removed, we had increased expression of CXCR2 primarily in our granulocytic population. We went forward to look at their ROS production as well, and we do see that while it's not to the levels of mature PMN, they do increase their, or primarily granulocytes, increase their ROS expression during this time frame as well. And again, we repeated this experiment, but instead of fraction 4, we collected our total fraction 3 population of our immature cells and to test their infiltration capacity. And like I said, originally, immature cells should not have CXCR2, which means they should not migrate. Here we show that these cells actually gain migratory capabilities, specifically not during the exact time frame as PMN, more towards the resolution phase of our treatment. Together, these suggest that they display PMN function, so they're likely turning from immature cells to some kind of effector cell. So when we got this data, we were like, okay, what's going on? So we looked in the literature, and we found that under chronic inflammatory conditions, myeloid-derived suppressor cells have also, which are another type of myeloid leukocyte, have also been found to arise during chronic inflammation. These cells are a heterogeneous population of granulocytic-like and monocytic-like cells that are terminally differentiated and they're immunosuppressive. MDSC are regulated by the same factors as mature PMN, such as IL-6 and GM-CSF, and express these same proteins. So it makes it very difficult in experiments to separate these two cell populations. The easiest way to do this is to test for T-cell suppression abilities. MDSC have been shown to be very, very suppressive in cancer situations and recently in inflammation, and they're being discovered in more and more pathological conditions. Generally, these cells are very immunosuppressive, but their biggest characteristic is they're immunosuppressive to T cells. Specifically, their most common role is the uptake or consumption of L-arginine, which is a non-essential amino acid, from the microenvironment, which interrupts the cell cycle needed for T-cell differentiation, as well as interrupts that expression of CD3 zeta receptor, which is needed for TCR signaling, the TCR signaling. They also express PD-L1, which is the PD-L1 ligand. It binds to PD-1 on the surface of T cells to initiate apoptosis. And they are also able to downregulate IL-2 receptor and the TCR receptor through production of peroxynitrate on the surface of T cells. So to test their T cell suppressive abilities, we use the T cell proliferation assay. To do this, healthy mice, the spleens of healthy mice were collected and digested to collect splenocytes. This was total splenocytes, but in this experiment, we're focused more on the T splenocytes. At the same time, colitis mice, remember our treatment model, their bone marrow were flushed and subjected to per-cause gradient separation. We isolated these cells, and they were then, after T splenocytes were CFSE stained, which is a green fluorescent dye, co-cultured together to test the suppression of T cells. The T splenocytes were then activated by CD3, CD28, which are antibodies against the CD3 zeta chain receptor needed for T cell activation, as well as the co-stimulatory molecule CD28. The CFSC stain, the idea of this is an esterase. So basically, once it gets into the cell, it becomes cleaved and it cannot exit. This is why it's good to measure proliferation. The only way this fluorescence can decrease is if the cells are proliferating. And this is what we use. The fluorescence readout is what we use to determine if these cells are inhibited or they're proliferating. So we let these cells culture for 4 days and collected them and analyzed them by flow cytometry. We showed that indeed, actually, and this is the beginning of the project that I got from Alex, we showed that indeed these cells beginning at the post-acute phase and lasting throughout inflammation recovery were potently suppressing T cell responses. So this was a really, really big finding because I think, we went in looking at PMN and we found that we have immunosuppressive cells with these inflammatory cells. The next problem we had, before we went further into that, we wanted to confirm that these cells were actually MDSC. MDSC are, as I mentioned earlier, most commonly known for their uptake of L-arginine and their expression of arginase 1. In healthy conditions, arginase 1 is primarily expressed in the liver as a component of the urea cycle. But under pathological conditions, arginase 1 can be induced by M2 macrophages in MDSC, or it can be induced in M2 macrophages in MDSC. Overall, it has been identified as a marker or it has been used as a marker to identify MDSC in circulation and tissues. So in my model, that's what we use it for. This is just a preliminary Western blot, hence the ugly beta-actin. But here we just simply show that even compared to the increased amount of protein, our cells in the beginning of resolution to the end, after the insult is removed, induce this arginase expression, suggesting or confirming for us at least that these cells are immunosuppressive and they express arginase, so we can call them MDSC now. So with MDSC, as I mentioned earlier, they're heterogeneous. They're a mixed population of monocytes and leukocytes. So we wanted to know if one cell was playing a role more than the other, or if they were both contributing. So, we wanted to figure out which population was primarily expanding, and if there were differences in their immunosuppressive capabilities. To do this, we did magnetic activated cell sorting of these populations, where the cells from 2 to 3 mice were collected and pulled and fluorescently labeled with the PE anti-Ly6G antibody, and then with the PE selection antibody, which then we added microbeads to tag our target cells, put this in the magnet for separation, where we then isolated separated at least our 2 MDSC populations for analysis. After separation, MDSC populations were used for T cell proliferation experiments separately, where we showed that in agreement with our previous papers, monocytes seem to demonstrate just basal level immunosuppressive activity. We believe that this may just be a natural role for them in the bone marrow, but when we look at our Ly6G positive population, these cells were not immunosuppressive from the beginning, but gradually during the post-acute phase and lasting throughout recovery, started to gain this immunosuppressive capability. This, coupled with the fact that our granulocytes were the primary population expanded, suggests that the expansion of these fraction 3 granulocytes suggests that these GMDSC play a role in colitis because they're expanding, or at least in the post-acute phase of colitis because they're expanding during this time frame. So the role of MDSC in inflammatory disease has been studied, but in colitis particular, it's still under investigation. In studies of autoimmune models, such as rheumatoid arthritis and in experimental autoimmune encephalitis, EAE, MDSC were found to alleviate symptoms. And as I mentioned, they have been found in the blood, spleens, and colons of ulcerative colitis patients and mice models, but are still under investigation for their role. In some studies, they show that adoptive transfer of colitis MDSC to other colitis mice reduced inflammation and facilitated colonic mucosal healing. But the role has recently become controversial as other studies have shown that they may contribute to inflammation, especially through their induction of pro-inflammatory T helper 17 cells. So we wanted to kind of get an idea of what these cells may be doing in our animal model. To do that, we first looked at the spleen, where we saw that massive expansion of granulocytes in the spleen, more so towards the recovery stage. This expansion tightly correlated with the overall decrease in CD3 positive T cells. And as I mentioned earlier, that's one of their mechanisms, is to downregulate that CD3 zeta chain. So based on this, we started to believe that these cells expanding in the spleens are actually MDSC as well. We then looked at the colon. To, again, try to figure out what these cells are doing in the tissue. And here, between the three, comparatively, we showed that we get expression or the presence of Ly6G positive, arginase 1 positive cells in the colon beginning at 9 days, but massively at between 12 to 18 days, that correlated not only with recovery morphology of the colon, but also cell proliferation and tissue repair markers, alpha-synuclein and Ki67, increased expression in the colon, together suggesting that these cells, MDSC, may be playing a role in the resolution phase of inflammation, specifically tissue repair in the colon. So at this point, I've answered the first two questions, and I wanted to try to talk about the last one. We didn't get to really investigate into it, but the literature shows, well, our study shows that, again, IL-17 was the main cytokine systemically present in these mice. In the literature, they showed that it's significantly elevated, that is found to already regulate PMN differentiation, activation, and recruitment, and induce secretion of MDSC stimulatory factors, especially IL-6 and GM-CSF. Its neutralization in chronic colitis patients actually exacerbated the disease. So all of these taken together suggest that IL-17 plays a more complex role than originally thought. Unfortunately, we didn't get to really investigate too much of that, but based on all of these data together, we came up with a few ideas or a few hypotheses. But overall, functionally enhanced PMN are induced as a mechanism to deal with the unresolved insult, but may further contribute to the development of colitis pathology. Furthermore, we think that GM-DSCs migrate in and impair the leukocyte responses, especially those of T cells, PMN, and M1 macrophages, which literature has shown they also inhibit, to counteract their exacerbated responses and help promote inflammation resolution. And as far as IL-17, we think that it probably, if it is the factor regulating this, it would act indirectly to produce PMN and MDSC, as it is systemically present and can travel to the bone marrow and induce IL-6 and GM-CSF in bone marrow, fibroblast, and stromal cells, which has been shown in the literature. So in conclusion, we believe that when mice become chronically inflamed, there is a reprogramming event. And what occurs is that under healthy conditions, we have a fraction 3 population that contains normal immature myeloid cells and monocytes or MDSC, as we showed they had that continuous suppressive activity, as well as our CXCR2 high PMN. But we think that under chronic inflammatory conditions, we get expansion of immunosuppressive GM-DSC that upregulates CXCR2 and functionally prime PMN that are even more efficient at killing pathogen and infiltration, as shown by the enhanced infiltration. We think that the PMN then migrate back to the inflamed area to help accelerate the pathogen clearance and try to promote resolution, while our GM-DSC migrate in to try to suppress the inflammatory response and allow for inflammation resolution. And that IL-17 is likely regulating this. Just briefly, I'd like to touch on some other projects that I was a part of or contributed to the lab. I have 3 second author publications or co-author publications in the European Journal of Immunology and PNAS. And with that, I would like to thank my PI for all of her help throughout these 4 years. I couldn't make it, and I really am appreciative. I would like to thank all of my committee members, Dr. Shu and Dr. Denning. Thank you for all your help and your guidance. Alex especially for the prelim data, and Jen for all of the help as well, Sean for all of the help, Mahathi, and everyone else in lab, and this is just for funding. And then just some moments of my best times here at GSU, and I'd really like to thank my now fiance for sticking through 4 years of craziness, and that's it. Thank you. Questions?